Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kanakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kanakin, on October 11th, we will be hosting our Dr. GPCR Symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person Adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of Adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I'm very excited to have with me Dr. Justin English. Justin, welcome. This is the 3rd time we're trying. 3rd time's a charm.
We're going to get it.
Absolutely. Very happy to have you here. So how about we start at the beginning? Would you please introduce yourself, tell us who you are and what do you do?
Yeah. Hi, I'm Justin English. I'm an assistant professor of biochemistry at the University of Utah. And I make toys for understanding how cells communicate information from outside to inside and understand where information is coming from in their environment, mostly through G protein couple receptors.
Obviously, nothing else. This is a Dr. GPCR podcast.
It is the most important protein family.
Of course it is.
We've all written that at least once.
Yeah, absolutely. I just noticed that our Google Calendar invite is called Justin English Dr. GPCR Podcast Take 2, which
No.
I didn't put it in. This is Google knowing that we've done it. So when did you first hear about GPCRs?
Oh, probably somewhere in molecular biology in college as an undergraduate. I had to study them and probably heard about them and took nothing away from that. But I think my first real appreciation from them was when I started rotating in Henrik Dolman's lab at UNC Chapel Hill in the Department of Pharmacology. He was focused very heavily on pheromone signaling pathways in yeast, and started explaining how these peptides that are secreted bind to this protein, and it just had sterile 2 was the name of it, and I didn't really know much more about it, and then slowly began to develop a better understanding of that as a graduate student, and then immediately pivoted not to do GPCRs in his lab at all, but I learned about them there.
I love it. I also heard about GPCRs for the first time in undergrad, and it was I think during all these podcast recordings, I've said it a couple of times. For those of you who listened to the podcast and have heard this before, it was a class with Michel Bouvier, and I got hooked right then and there. And I actually wanted to do a summer internship in Michel's lab right after that class because it was March or April, the early 2000s. I didn't end up going with Michel, but I was hooked. That was it for me.
That's awesome.
So how about you? So you mentioned doing a rotation in Henrik Dolman's lab at UNC and then ending up not working on GPCRs. When did you start working on GPCRs?
I mean, I didn't really actually start working them in earnest until I was a postdoc in Brian Roth's lab. Almost throughout my entire scientific career, I just kind of went around to different signaling systems. And so I started working in yeast and like mismatch repair at Cornell when I was an undergraduate. And then I went to the NCI and I was working on like P53 and animal cancer models. And then I worked with Vicky Bouch at UNC Chapel Hill on vascularization of the eye and how VEGFR like signals. So I've always been really interested in how cells receive and respond to contextual environmental information. And it wasn't really until I started working in the pharmacology department at UNC that I appreciated that most of the ways that cells respond to their environment are through GPCRs. And I didn't really start working on them in earnest until I was a postdoc. Yeah.
Interesting. So let's go back to the beginning. Did you always know you wanted to be a scientist?
Oh man, no, not until 6th grade, I guess. There was this one point when I was growing up, right around like when I was a sophomore in high school or something. I told my dad I wanted to be a scientist. And he immediately hired a career counselor because he thought that was a terrible decision. And he wanted me to take all these tests to assure myself that I wanted to be a scientist. So while I was taking the test, I would just answer the questions however I thought the most like Poindexter scientist would. Do you like taking notes and keeping like careful records? Yes, absolutely. Like, yeah, what sophomore in high school loves doing that? You know, there are some, but I certainly knew I really liked learning about the natural world and how things work. And so right around 6th grade, I started doing Science Olympiad with this high school teacher named Mr. Owen. And he was like my Science Olympiad coach for many years when I was in middle school. And that's what really got me into like scientific investigation and trying to understand how things work. And it kind of went from there. I've always loved it after that.
Was it more a biology focused or more chemistry or physics focused?
Very biology focused. Cutting up plants, putting like membranes on slides. I mean, when I looked at my first cut up leaf under a microscope, I was like, all right, well, this is pretty great. This is it. This is how things work, and we're gonna figure this out. That's all I want to do. All biology all the time.
It's beautiful. It's actually beautiful. When I think about looking at cells under a microscope and then you get to know your cells, obviously, in my case, it was more hex cells than anything else, but I could tell if it was a good day or a bad day to run the experiment just by the way the light would shine through the cells.
Yeah. It's very tactile in a way.
Yeah. Yeah. Oh. So you're in 6th grade, and then you get to high school, you do all these tests, and then what gets you to where you are today?
I was an okay student in high school. I got deferred from 2 years from Cornell University where I wanted to do genetics because my grades weren't particularly good. To all high schoolers out there listening, I feel your pain out there. I was not very good at Spanish or English or history, but I went to SUNY Binghamton, which is like a state University of New York program in New York, and I studied biology and chemistry there, and then transferred to Cornell University. I worked with this guy named Eric Villani in the molecular biology program there, studying yeast, and I thought that this was just the coolest system, that you could do genetics and knock genes out and understand how certain things worked. And we were trying to study how mismatch repair in non-human polyposis colorectal cancer was mediated by mismatch repair damaged genes. And I got so into it because I felt like this is how you solve problems in human health, is to really like figure out how all these parts work. And I left that and started the NIH post-baccalaureate program at NCI immediately after, and spent a year in Bethesda working in the NCI and working with animal models and really kind of developing a sense of what all these proteins do and how they communicate. And then I applied for graduate school and ended up going there were two decisions, and I just picked UNC Chapel Hill. I thought it sounded like a really great place to be. It was an umbrella program. I didn't like pick the pharmacology department even at that point. I was the first year of students coming into that program where you just there was no department. You just kind of came with a group of several hundred other interested scientists, and then you just found your way. And then how did I get into the pharmacology department? I did this thing that I would come to regret, which was that I read the scientific biographies of every professor working at UNC Chapel Hill. I wrote down all the people I thought sounded like were doing interesting science, and then I sent out an email to all of them in one big blanket email. I blind carbon copied everyone, mind you, but it turns out professors talk to one another. I didn't know this at the time. And I sent out an email to like some 120 people saying, hey, your research sounds really cool. I have 3 questions. Are you still doing what's on your webpage? Are you interested in the topic that you have like grants on right now? And are you accepting graduate students? And one of the people that wrote me back was Henrik Dolman in the pharmacology department. He said, why don't you come in? We'll talk about, science and maybe how you can do this better in the future. And I said, that sounds great.
I love it. I love it. Well, yeah, you were efficient. Come on.
I was just trying to save time. There were so many interesting projects. I didn't know how to figure out how to contact everybody.
Yeah. It was one email instead of 120.
Yeah. I always like doing things in high throughput whenever I can.
I love it. And then, so you're at UNC. You're working in Hendrik's lab, and then what? Did you always know that you actually wanted to do a PhD and then a postdoc, or was there a period there where you were thinking, am I doing the right thing here?
Yeah, that's frequently. I still ask myself that question sometimes. I say, what am I doing? Is this the right thing for me?
Good question.
Yeah, it's always, I think it's good to evaluate your goals. I think somewhere around the time I did like my 100th Western blot in Hendrik's lab, I was really questioning if this was for me, but I pushed through. I think I always knew I wanted to do science, and I felt like the route to getting into being a professional scientist was to go and get my PhD. That was what a lot of my mentors at the time when I was an undergraduate had mentioned that I really needed to do if I wanted to understand it. And I think that's right. I think that without having done my PhD, I wouldn't have come to a full appreciation of how to really think through scientific problems and begin to address how to come up with solutions for them. And so I think that was the right choice. And then the postdoc, at the time when I graduated, I hadn't really thought very much about what happens after I graduate. Sort of tunnel visioned into like, you're just going to get this PhD. And then, I hadn't put as much thought as I really wished that I had at the time about how I wanted to structure things. But, I went to seminars all the time. I was always asking questions. I was really excited about what people were presenting. And then my PhD was done. I had defended. And then at one point, my friend Kate White, who worked in Brian Ross' lab upstairs, Henrik was on the 3rd floor of the same building as Brian, who was on the 4th floor. They were in the same department floor. My friend Kate White messaged me and said, hey, are you looking for something to do after you graduate? Yeah,
just in case.
I think Brian's looking for somebody. You should ask him. And I came up and he said, you always ask really good questions in seminars, and I think you'd be a really good person to have in the lab. Why don't you join the lab? And I was like, okay, sure. This sounds great. You're a really good lab and a really good scientist. I think that would be a really great opportunity. And so I accepted. I didn't interview anywhere else for postdocs. I was just happy that I was going to get to stay in the same place and get to work on cool projects. And he and I kind of talked about some visions and ideas I had for stuff I wanted to develop, and it very quickly turned into 5 years of working there, which was
I
fun.
love it. You just had to move one floor up.
Floor up, yeah. Yeah, it was great.
Joking aside, it is very important to go out and go to seminars and listen to what other people are doing and asking questions. And I think, at least I know from when I was a student, I didn't ask as many questions as I wanted. And a lot of people who listen to the podcast are between 25 to 35, so PhD postdocs. And I want to take a moment to talk about how important it is to be able to put yourself out there and ask questions. And you just told us that allowed you to just say, "Hi, Brian. I hear you're looking for someone. Can we talk?"
Yeah. I was always scared to ask questions, actually, and so I started forcing myself to go to the meetings. And then when someone would say something, I couldn't ever remember what my question was. They'd be having the seminar and I'd be listening and I'd think of a question and then I would forget it by the end of the seminar, or something that I was interested in. So I'd always bring a pad of paper. And as they were going through, I would sit and take notes about the questions that I had and what they were talking about. And then I would come back to them. I just made it a rule that I had to ask one of my questions, if I had one. I try not to say a comment or
Yeah, great talk. Thank you.
no, but if I have a question, I tried to force myself to ask the question that I had, even if I felt like, I should know this or something else. And then I found that I actually really enjoyed it, because I liked thinking harder about what it was that I was really trying to ask, and it helped me to really refine how I talk to other people about what I don't understand. And so I felt that was pretty helpful. But yeah, I really had to force myself. I was very nervous to do it when I first started.
Any advice for people? Bring a pad, force yourself. What else?
I think it's always true that if you have a question, you didn't understand something, the majority of people in the room didn't either. And a lot of people aren't comfortable asking that question, but if you feel like you really came away not understanding something fundamental, it's one, every speaker, no matter who they are, wants questions at the end of their talk. If they give a talk and there are no questions at the end, that is the worst feeling at the end of a talk. So they are excited for you to ask these questions, especially easy ones like, can you explain the core component that I didn't understand, right? That's a low ball, easy one for them anyway. So you should always ask it. They always want questions. But I think, if you really feel like you don't understand it, nobody understands everything. And I always tell my students this as well. Just because you think you should know it because other people seem to be understanding it doesn't mean that the majority of people know what anyone's talking about. It's the same with giving a good talk. You really need to presume most people don't really know what you're trying to communicate, and to make it very obvious and transparent what the problems are. And if you feel like you didn't grasp that, most people are probably also wondering the same thing.
Yeah. And I think, and then to your point, when giving a talk, it's important to think about the audience, because you're an expert at what you're talking about, but not everybody who's going to be in the audience knows what you're talking about. So you need to explain and tell a story behind it. And I think that's the extra effort.
Yeah.
Being an expert specifically at what you're doing, yes, it's a lot of work, but you can do it. But being able to get out of that, let's say, like that bucket of expertise, and from a high level explain to others who are not experts, who are not in your field, why is it important what you're doing? That's an art.
Yeah. Or even people who are experts in you. I can't tell you how many times I go to a talk and someone glosses over the difference between two different regulatory proteins, like GRK1 and GRK2. And I'm like, I actually don't quite remember what we know about those two things and what the differences are. If I could just get a quick brush up on that real quick. I'm going to learn at the phosphorylation meeting next week. That's going to be a critical meeting. But, people get really focused on their topic, right? That you're thinking about every day. And then everything else sort of, if you're not constantly refreshing your memory about it, when somebody pops it up, even something that tangibly you should be pretty familiar with, a little refresher about what these things are and why we think they're important is always important.
Absolutely. So what did you work on in Brian's lab?
Cool development.
what did you want to work on, and what did you end up working on? I guess that's all. That's because
I always joke that we had day projects and night projects in the Roth Lab. There was always money to do whatever you wanted. The thing that really was limiting was the time. And so I would have projects I worked on at night that were just out there, crazy. I remember at one point we were on like a patch clamp rig trying to make a magnetic calcium sensor, which actually now I think we're darn close to making, and it's other laboratories have been working on it, but we like spooled a electromagnet that we put underneath like a plate rig and then turned it on and oscillated it. We were trying to use all these proteins that were supposed to bind to ferritin and like drag it to change the conformation. Wow. Anything, like everything that we could think of where we wanted to get like greater control over our ability to read out and control cellular signaling from GPCRs. We tried everything. So I was on a brain initiative project in Brian's lab where we were developing some of the new like DREAD ligands and DREAD components, and we published one of these for deschloroclozapine in primates, and that was a really fun story and an exciting project that I got to be a part of. But it was, a huge consortium project with folks in Japan. Development of biosensors like the TruePath system, which is like a kind of an expansion of Michel Bouvier's really great work and making sensors for alpha, beta, gammas and understanding their dissociation. That was my friend Reed Olson and Jeff DeBerto really spearheaded that, and I got to help a lot with the molecular cloning and the like large scale development of those tools. How do we measure dissociation? I developed this platform called Vegas, which is a directed evolution system, but mostly out of like desperate need. During the time when I was there, we were getting all of these structures of G protein, the 5-HT2B receptor bound to LSD, the dopamine D2 crystal structures and cryo-EM structures. And part of that was nanobodies were like this big thing at that time, right, for stabilizing the conformation of the heterotrimeric G proteins. And I had this idea that we'd use directed evolution to try and make nanobodies that would stabilize these receptors and these conformations. And we had Daniel Wacker, who's now at Mount Sinai, was there, and he had purified 5-HT2A and put LSD in it. And we had injected these llamas, and we're glad that they weren't all hallucinating because there was like LSD in the mix. And getting the nanobody libraries out and then trying to evolve them to basically stabilize the cytoplasmic face of the receptors. And we actually were never successful at getting the nanobodies to stabilize a single conformation that we could see in cryo-EM, but in the process developed this platform for the directed evolution of proteins that could allosterically modulate receptors and change proteins in really fundamental ways for directed applications. And a whole slew of other, I've made probably over 14 different Cree recombinant animal lines there for the Illuminating the Druggable Genome project, where we took orphan receptors and we went to the end of the individual exonic locus and we cut it out, and then we put an inducible P2A Cree after, so that we could have Cree mice that were expressing Cree in the cells that were expressing the GPCRs. We did all sorts of. It was like one of the best times. I mean, like everybody in their postdocs right now, maybe people watching this. Postdocs are hard, but man, once you're out of it, you look back on it and think, man, that was awesome. What a great time.
Yeah.
You have so much scientific freedom and so much time to explore so many, really, anything that excites you. It's such a, it's something I miss a lot. It's really fun.
It can be a very prolific time. When I was defending my thesis, it was Ali Salapour who was on my committee. He was in Michel's lab doing a PhD at the time, that did a postdoc with Mark Curran at UNC. And then he's a professor at the University of Toronto. And I remember Ali at the time, he said, Yamina, you're going for a postdoc at Rockefeller. Have fun, because after that, it's over. After that, you have to actually write grants and get money to do whatever you want to do.
It does. You start to take your hands off the pulse of what makes it the most exciting part, right? Like that is the daily work of really making discovery happen. It's so exhilarating in a lot of really fun ways.
Did you always know during or after your postdoc that you wanted to become a professor?
You know, I thought about it a lot. It was actually kind of a weird time. I was going out for interviews in 2019, right in the fall,
Yeah.
and then right
What
around
a good
2020,
something happened.
yeah, some stuff happened. But most of the places that I was really looking to go couldn't give offers uh, to come. And so really there's only one place left to go. It was here at Utah. That was the only remaining place that had an offer available. And I spent a lot of time thinking about, like, all right, is this really what I want to do? Am I going to go here? Is this what I'm going to do? This is the place I want to be. And it worked out. But it was kind of a gamble at the time when I really thought about it. I'd never really been here to Salt Lake City. My wife had never been. She didn't get it. We didn't do second interviews. It was one interview, and then I was just like, well, I guess we're going to take it. And we drove from North Carolina to here in an RV because it was the middle of COVID. And yeah, it worked out. But I had a lot of soul searching about if this is really the commitment that I want to make to go forward with my career. And it's been a lot of fun since we got here, but it's also been a lot of challenge. Just partly starting a lab, starting in that kind of environment during that time. But I think I wasn't sure, and then I kind of got to the end of my postdoc and started thinking about the same as my graduate school. Like, what is going to be the next step here? And I really like doing my own independent research, so I was really excited about that possibility. But I think, there's been pros and cons to the professorship, and I don't really know what the other side of that coin looks like going to industry. I haven't experienced that, and so I can't really compare and contrast them too closely.
Did it occur to you to explore industry, or since this offer came in and you decided, okay, this is anyway what I want to do and let's see what happens?
Yeah, part of the reason that I ended up coming here is because it had a very industry-friendly sort of mindset. A lot of companies are spun out. It has really good agreements on patents and licensing. So I already actually have my own startup company here. For some of the technologies that I've developed for the Vegas platform that my lab works on. But I really like both, and I found that I really enjoy both. So we've done a lot of venture capital work and raising funding for the company, deciding on strategic initiatives for, basically how we're going to develop this company into something that's going to be profitable and that people are going to be interested in with. I do a lot of sponsored research agreements. I work with Eli Lilly pretty extensively right now. And so it's fun to have an academic lab, but I really actually am enjoying this interface with the industry-facing component of pharmacology quite a bit.
I love it. No, this is great. And I'm also happy that I'm not the only one who was in this position of finishing a PhD and be like, okay, so now what? And then finish a postdoc and be like, okay, so now what? That's why I had to do, not exactly why, but part of the reason of me doing a second postdoc was exactly this, is I realized it too late that I wasn't ready I didn't have a plan and I had to do something.
Yeah.
And it turned out to be a great opportunity because I was actually at NIH as well, on the main campus at NIAID, and this was 2016 to 2019. And it was a great experience. I got to do a little bit of animal work on different GPCRs, and that's when I was like, okay, I'm done with academia. I don't like to write grants. I need something else. And that's what got me to the other side of the coin, which is a very interesting side.
Yeah.
I think the scientific rigor is there. Is absolutely there. And if you find the right team, the right company, the right position for you, it can be exhilarating and a lot of fun as well.
Yeah. Yeah. It's fun to see how quickly things work over on that side of the fence. We're here and then something is going well and you pass it off and then very quickly that turns into something else. It's fun. It's fun to see. And I really like being able to keep kind of my feet in both sides of things right now.
And that's very unique, I have to say. I don't think we've had a lot of guests on the podcast who are able to do a little bit of both, and being able to work as an academic, but also have these business relationships with different companies, start their own company. It's a very unique position you're in. It's really cool.
Yeah, I never follow any blueprints or plans. I just kind of do what I think is the most fun thing at the time. And if something presents itself and I'm like, that sounds fun, I go for it most of the time. That's usually what drives like 90% of my decision making. I'm not as strategic as I might seem. I just think like, that sounds great, let's do it. And then I just go for that thing. So yeah.
I love it. So what's your favorite GPCR?
Oh my gosh, that's like asking which one of my children is my favorite.
Could be a family. I'll take a family of GPCRs or accessory proteins. Doesn't have to be one. You can rank them to a top 3.
I mean, I think that I'm really interested in the pH sensory G protein couple receptors now. I get to help Ashish Manglik and Willow Coyote Mestis with a really great paper that they have coming out that's on bioRxiv right now on trying to elucidate the mechanisms of how they detect these kinds of changes in pH. But I think we're going to continue to find like these really interesting evolutionarily conserved mechanisms for detecting environmental stimuli that aren't necessarily like orthosteric pocket, but binding in sort of a class A like but distal pocket effect. And I think those are really cool. The others are the SREBs, which have vexed me through my entire postdoc and professorship, like GPR85. These are receptors that have never changed almost a single nucleotide since bony fish. They have single exons. They are the exact same amino acid sequence as they were back then. They're like these primordial, neuronally expressed GPCRs for which we have just absolutely no real idea what they're doing. And they've been, when you delete them or overexpress them, they change brain morphology and shape and size, and they're everywhere. And we haven't been able to figure them out. What do they couple to? We don't know. Do we have really good ligands for them? There are a bunch of people that have been working really hard on that. We don't really have really great bona fide ligands for them yet. I would love to see those solved, just for the excitement of like, why are they conserved and what's going on with them and what do they do? What's their mechanism? And I think those are probably also my favorite.
So you love a good challenge.
I love a good challenge. I love a good puzzle. I want to know how we can develop things that kind of open up new avenues of investigation. I always joke that the way that I see my role in science is to come to like bare cliffs and make footholds for other people. I'm not sure that I'm a great explorer, but I am really good at banging my head against the wall. And I like doing it and making tools to like solve that problem. So yeah.
I love it. So when you started your lab, you mentioned, it was COVID and it was difficult to recruit. And how big is your lab right now? And how did you set it up? How did you go about setting it up?
Yeah, right now we actually just lost our bioinformaticians because they're tough to hold on to. But before they left, we had 10 people in the lab. We have 2 senior people, a lab manager, Katie, and a research associate, Adam. And then I have 5 graduate students. And then we had 2 master students who were computer science majors who were doing bioinformatics, Sam and Caleb. Both of them have now moved on, but the goal is to try and synthesize good sort of senior technical hands who are then assisting graduate students with independent projects that they're developing that all kind of circle around molecular biology and pharmacology, and then bioinformaticians who are working with those groups to try and develop tools and pipelines for analyzing their data as it comes out. And so that's mostly how it's organized. I'm involved as well.
Really? Didn't sound like it. I'm kidding.
Yeah, I'm in there, and yeah, so it's student heavy, but with technical experts who are there to try and help guide people and aid them when they get stuck.
I love it. Yeah, I think having a good technical experts in your lab is just priceless because these people really know their thing really well. They could do this in their sleep.
They're
and every time right? Every time they do it, it works exactly the same way. And you're like, wow.
Yeah.
And it's just magic.
Yeah, and a lot of really good, creative conversations from people who have been thinking about these kinds of problems for longer. I would really love to find a postdoc, but I think right now the landscape is really tight and competitive for great postdocs. I think less people are going into the position now than historically they have been, and the costs are very high. And so you really need to find a good fit. I've interviewed several people who have been interested, and often I have conversations about what are your goals and what is it that you would like to do after the postdoc, and how can we minimize the time that you're a postdoc, because it's not really that desirable of a position, and
Yeah.
I think the whole landscape for that has really changed over, maybe pre-COVID, post-COVID. And so we continue to look, but we haven't found great matches yet, but we will hopefully soon.
As soon as they're going to listen to this podcast episode, you're going to have a flood of emails coming in and saying, now I know I'm going to come and join you.
Sure.
Well, if anyone's looking actually for a postdoc and they want to reach out to you, they will be able to do so. They can already do it, but we're going to have this episode on the website with your contact information as well. So
great.
It's going to be really cool. All right. So you mentioned you're developing tools in the lab. Can I ask you what can you tell us about these tools? I'm intrigued.
Sure. Yeah. We published a few. I worked with my friend Fleur Ferguson at UCSD on coming up with a method for directly biotinylating drug-bound complexes. So taking drugs that would go into the cytoplasm and bind to a particular drug, and then have an inducible biotin ligase that labels the proteins that the drug is bound to, and then using cold competition to remove it. That's sort of an open-ended question around. I like to think about a lot of the tools that we build as trying to answer three key questions. What are the inputs and the outputs and the signal processing that's happening when any kind of pharmacological stimulus hits a cell? How do you solve those problems? So input would be, what are they binding to? Who are they interacting with? Who are they talking to? What's happening? Outputs, we have a really nice bioarchive paper that we're going to be publishing soon on synthetic transcriptional response elements. We made this large library of really high affinity transcriptional response elements that are very small promoters. They're some of the strongest promoters that respond to endogenous transcription factors around, and they're only about 200 to 250 base pairs in size. So they're very tiny. And when you stimulate the cell, the transcription factor that's active goes and binds and activates. Very similar to CRE and SRE, but we made a very large library that you can do in parallel using what's called a massively parallel reporter assay, where a bunch of barcodes are activated and tell you which of these several thousand transcription factor binding elements have been activated by a stimulus. I found all sorts of really fun and interesting things in there. We screened a bunch of G protein couple receptors and demonstrated which things are actually downstream of certain alphas like GQ. Turns out it's not SRE or THRB or a lot of things that people have canonically looked at to say like this is downstream of GQ, but NF-kappa B1 is, at least in HEK cells, it changes from cell to cell. It's like playing whack-a-mole with molecular pharmacology. And so we're using Vegas pretty extensively now to try and evolve nanobodies to be allosteric modulators of G protein couple receptors and then make them optogenetic. So one of the goals is to deliver them to cells and then have on-demand control of when G protein couple receptors either signal more or less from their endogenous ligands. I like to think about it a lot as your body's really kind of playing a song of ligands, and they release them in very specific tempos and behaviors. And so how do we move the volume knob on that up and down in a controlled way in a given cell type? And so one of the goals is to make allosteric modulators that you have a lot of control over inside of a given cell to say, I want more signaling from this receptor or less signaling. So that's a class of tools that we're currently developing, along with a suite of other accessories for directed evolution. Currently, we have a fun paper coming out on high fidelity single molecule sequencing on nanopore. So how do you get a single strand of DNA to give you its exact true sequence in a pool of millions of unique sequences without needing to essentially do Illumina sequencing and then use that to register on your nanopore sequencing, which is kind of the gold standard currently. Yeah, how do we solve major problems in molecular pharmacology that will let us try and zero in on what we're doing? We have new developments with the Vegas platform. We've been working with Willow's group on how do we adapt our TREs to be better signal processors for deep mutational scanning. We have fun web tools. If you go to our GitHub, where you can look at all the data that we've generated and kind of browse through how we think about displaying and thinking about really large data sets that capture more depth in reporters and pharmacological mutations of G protein couple receptors. But that's like sort of a flavor for some of the things that we're really interested in. You'll notice I didn't mention a receptor in there.
No, because you're developing tools. You want to be able to look at any receptor you want to look at.
Yeah. The goal is to make something that is multimodal for any target that the people out here listening are interested in.
Exactly.
So your favorite thing, we'll send them to you. You can run whatever you'd like on whatever target you think is most exciting to you.
I love it. So you mentioned starting a company. Can you tell us a little bit more about that? I'm curious.
Yeah. So the Vegas system is quite powerful, but we're really trying to bring money in to leverage that to make pharmaceuticals. For the most part, the platform itself isn't capable of doing that. And so while you can evolve things in the cytoplasm to bind to other things, nobody wants a peptide that needs to work in the cytoplasm as a drug delivery agent. And so how do we do things like get peptides on the extracellular surface of our drug targets and then directly engage with them to then select for that peptide that then will be functionally active? Or how do we make allosteric modulators that are going to stabilize receptors and other targets in their drug favorable conformations? And what are we going to do with those? And so when I came here, there's this really great Utah Therapeutic Accelerator program that its central goal is to try and identify technologies and discoveries at the university and move them into commercial spaces. And there's a guy named David Beers who heads that up here. He knew a bunch of people who were looking to start companies in the area and put us in touch. And it really, being at a university that is in an industry-focused mindset really helps to facilitate that. If there are really good commercial offices that are going to handle and manage your patents. We're about to file our 3rd and 4th patents since I came here this month on a bunch of technologies that we've developed in the lab. But if you're in an environment that values that and that they fund and push that forward, it makes it a lot easier. I never imagined that I would do that because I felt like I'd never have time. But then the teams to manage it sort of came together and it made it much easier for me.
You get that support. I'm mindful of the time, so let's start inspiring young scientists.
Yeah.
Let me ask you this: top 3 aha moments that changed or shaped your trajectory.
I think the first one was when I was sequencing Simbus virions that I had been evolving and actually saw sequences that matched known functional consequences and thought, oh, this is actually maybe working. This is great. And that was maybe not more aha, but pretty exciting. I feel like I don't have a lot of aha moments because my aha moments are more like, it's working, rather than discovering something fundamentally new. But to see the data come off of the things that I've built and recognize that this is the indicator that we're going in the right direction, we need to continue to develop this. So that was pretty exciting. I would say when I was working really closely with Tao Che, who's now at St. Louis, but he was in Brian's lab, and we were working with nanobody 6. It was like from Jan Steyert's group. They had taken a kappa opioid receptor and panned it and gotten nanobodies that bound to the kappa opioid receptor, and it seemed like they were binding them in drug-dependent states that were selected to active and interactive forms. And we were on the microscope trying to use these to basically visualize active and inactive forms of the receptor. And we would put the drug on, and the nanobodies all went right to the plasma membrane and bound to the receptor, and then we'd antagonize them, they'd all go away. And I thought, there's something to these nanobody things. These are really cool. Like, how neat this state selectivity. Of course, I already knew this, right? Brian Kubilka had already had the heterotrimeric crystal structure. We all knew that nanobodies are stabilizing these states, but to see them be able to dynamically come on and off of the receptors when they're actually changing their ligand bound states was really exciting at the time when we were running those experiments. This was, gosh, I don't even remember. It was probably like 2015 or 16 or something like that. And it was really fun. It was a really exciting time. And then let's think. I think probably from my own lab now, we've been running this TRE-EMPRA platform for a while, looking at unique TREs from different G protein couple receptors. And we're working with Ruth Huttenheim's group, and they sent us a receptor that they're really interested in. And we ran it in the platform, and a totally different TRE popped out than any other GPCR is activated in the same cell background. And got really excited about that, seeing that like this tool set that we developed basically as a discovery mechanism could rapidly identify sort of non-canonical signaling mechanisms of receptors that have been studied pretty extensively, but never shown to actually couple to the pathway that we were identifying. And so that was really exciting and made me feel like we were making systems that actually had fundamental value in discovery.
That is
Great.
really cool. I mean, the nanobody experiment that you just mentioned, it's really cool because when you think about different conformations, you can imagine it, but seeing a consequence of that live is just magnificent.
It's really cool. It's really cool. And being able to reverse it and see those things come up.
Of course.
It's really exciting.
I love it. Any advice for junior scientists who aspire to contribute to the field?
Man,
that's a tough one. Well, bring a pad and paper and a pen and ask questions. I'm keeping track of it. Well, because the last question here is, what should we title this episode? So let's start thinking about that. This is a new question we're introducing. You're the first one who I'm asking this question. But any additional advice?
I mean, I think as you're getting going, do the things that you're having the most fun doing. I think if you're having fun and you're passionate about what you're working on, and you really think that it is an exciting thing to be working on, I think that is probably the most important thing to keep in focus. And when you're not having fun, assess why you're not having fun and why the thing that you're working on isn't exciting to you anymore, and try and figure out what is exciting and how you can work on that. Not because the project is hard. The projects are always hard. But if you're having fun and the project's hard, I think that's great. I think if you've lost your excitement to work on whatever it is that you're doing, then you really need to take a step back and ask, what is it about it that isn't contributing to that anymore? And I think good self-assessment is going to lead you down the path of what brings you the most satisfaction in your research. And I think that leads to all the other roads of success in a lot of ways.
Yeah, no, I like it. And I was going to just mention that and this goes for industry and academia and everything, and every part of your life, self-assessment and pausing and asking, am I in the right place? Am I doing the right thing? Is important to do it regularly.
For yourself.
Yeah, of course.
Yeah.
Yeah.
Some people ask that of for other people, but I think it's mostly for yourself. Yeah.
Yes. Yeah.
And I think that has really been what's driven a lot of the decisions that I've made in my career. I just look for the thing where I feel most excited and say, I think that's what I should be doing, because that's what makes me excited, makes me want to get up every morning and really grind at this. It's not easy. It's hard. It's so hard.
Yeah.
And so if you're not excited to do it, it becomes just unbearable,
Yeah.
So you have to feel excited every day to get in there and hit it.
Absolutely. Absolutely. So last but not least, what should we call this episode? Because we talked about all sorts of things, and let me just maybe kind of summarize what came through.
Yeah.
You're interested in making tools to better understand how outside context-specific stimuli act on cells and what the consequences of those are.
Yeah.
You plan ahead. You just kind of went from experience to experience with the focus of, is this something that I have an interest in doing?
Yeah.
If it's fun and you want to do it, just go for it. What else? Your team is built out and you are part of your team in a way that you do have, these very good technical hands. You have a team of young scientists. You have also the back end on the data side to help analyze the data output of all of these tools. And you also have the opportunity to work with industry and academia and help the GPCR field get these tools and use them with their favorite receptors.
Yeah.
So what should we title this episode?
Having fun helping out everyone else.
I love it.
Yeah.
Or the other thought I had is, come help yourself with our tools type of.
I mean, the greatest joy that I get out of the entire enterprise is seeing other people pick up what we've made and apply it and have success. I think that is, for me, really the mark of success for me and for what I do, is to see that what I've developed is really accelerating discovery for other people and making it possible for them to access problems that were unachievable or difficult. Yeah, I think that is maybe what I'm most excited in.
I love it. I think we just found our title. I'll let our team take this last clip and come up with it and condense that, because I think that we just found it. It was awesome talking to you. Thank you so much for your time.
I appreciate it. Thank you. You guys have a good day.
I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.